Antonia. So thank you for all the fantastic talks. I'm really excited to be chairing the very last session of a great trainee symposium that is focused on advances in behavioral models and experimental treatments. And our first speaker is John Hernandez, who is a postdoctoral research associate in Carla Cowan's lab at Brown University in Rhode Island. And he will be talking about an exciting new operant-based paradigm for examining motivated responses in flies. So take it away, John. I'll give you a 2-minute warning.
Okay, great. All right, okay. So good afternoon, everybody, ladies and gentlemen, and all those that I identify in between. My name is John Hernandez. I'm a postdoc in Dr. Carla Cowan's lab. And I'm really excited. Thank you for the committee for allowing me to talk about my work. What I'm really interested in is investigating how does individuality in alcohol preference manifest. But before I begin, I think it's important to just take a second and ask ourselves. You don't actually have to answer, but why do you drink alcohol? You know, people maybe drink because of past experiences, stress, for many because it's 2020, right? There's a whole host of reasons why individuals choose to drink alcohol, which makes it very difficult to pinpoint the biological underpinnings of alcohol preference. So I was very blessed in joining the Cowan lab, where I'm using a really neat operant assay that is cleverly coined the open bar arena to ask this particular question. So in this depiction here, there are 6 arenas here. They're about 4 inches long, and there's a camera above them. And so how this system works is it works based off of a Python interface. I basically put tape below half of the arenas that are, in this case, yellow. And when I put flies in, I selectively choose that area and basically tell the Python code, hey, when a fly enters this area, they're able, which I coined the ROSA, or the region of self-administration. When flies enter this area, they're able to self-administer volatilized odors or drug rewards, which for the case of this talk, I'm talking about 50% ethanol. This all works based off of an Arduino based solenoid system. So the exact second that a fly walks into this arena, there's volatilized rewards administered to them. And for the purposes of my studies, I study both male and female flies. All flies are virgins, which is important because we do know that sexual experience has an impact on ethanol drinking. These flies are given 15 minutes per session to self-administer rewards, and this is done for 3 consecutive days. And hopefully what you can appreciate from that is that we get to look at the overall trajectory that an individual fly can self-administer rewards. So one of the cool things about the data sets that I've collected from here is that there is pretty robust variability in at least 50% ethanol self-administration. So to orient you, the y-axis, each row here is one individual fly. And you can track that individual fly's behavioral self-administration patterns across 3 days. The color blue boxes denote flies that spend very, very little time self-administering. And the yellowmost boxes denote flies that have spent the entire time, and these are in 30-second bins, the entire 30-second bin self-administering rewards. And I think what's really cool that you can see from this data set is that you can see that there are roughly 3 at bare minimum populations of flies, right? There are flies that on day 1 self-administer very little, and then as the days progress, they self-administer more, 50% ethanol. There are flies that are relatively stable. They self-administer roughly the same amount for the same amount of time from day 1 to day 2 to day 3. And then there's a population that self-administers a decent amount on day 1, but they slowly decrease their self-administration. So I've coined these escalators, stable drinkers, and de-escalators. What's very interesting is, so this data set is just 50% ethanol, but I've also run flies for appetitive natural rewards. And so what hopefully you can appreciate from this is that the overall distribution of these populations of flies, escalators, de-escalators, and stable drinkers is relatively similar between apple cider vinegar, which is an innately appetitive odor, and 3% ethanol, which we happen to know it occurs in nature in rotting fruit, right? But this is where I think things get really cool, right? So when we look at the distributions for 50% ethanol, I think one of the most glaring things that I noticed is that the frequency of escalators in the 50% ethanol group is at bare minimum 2 times that of what you see in these appetitive odors, right? Potentially speaking to the pharmacological action that alcohol or ethanol is having in the brain of these flies. But another thing that I noticed in this data set is that overall, of course, if you look at the population as a whole, they prefer apple cider vinegar more than 3% ethanol, and they prefer 3% ethanol more than 50% ethanol. So I think what this operant assay allows me to do is very much ask that question, is in a population of flies, if we look at, let's say, just escalator flies, what is the distribution of time that they spend self-administering appetitive and drug rewards? Interestingly, they spend significantly less time self-administering 50% ethanol than they would for apple cider vinegar or 3% ethanol. But this is where it really got me excited, because when I do this same kind of analysis where I pull de-escalating or stable flies, that this overall hierarchy of drug or odor preference is actually the same between the subpopulations. So what this potentially suggests is that at baseline, without considering the phenotype of self-administration of individual flies, there's an almost kind of hard-coded innate preference for odors, right? But you may be looking at this data and saying, well, but John, you have an open bar assay that can do so much more. You can look at motivation behaviors. Well, you're correct. I think we can do much better with these data. So one of the really neat behaviors that I notice is that flies can actually, spend some time walking around the non-rosa area, and then they can enter the rosa and they can leave, right? So functionally, this allows me to analyze more subtle behaviors associated with drug self-administration. For example, the average time per bout that they spend self-administering these odors, right? And what I noticed is that when I look at this motivational behavior, that for 50% ethanol, they spend significantly more time per bout self-administering ethanol, which is really cool. But then I wanted to look at, well, what's the average latency? So if a fly leaves the rosa and then decides to come back to self-administer again, what is the average latency for this behavior across the odors or drug rewards? And what I noticed is that for 50% ethanol, the escalating flies spend significantly less time after they've left the rosa to re-enter, right? So all these behavior together suggests that what we really need to do is understand the more subtleties in these behaviors. So what I'm using is a C-Trax Java machine learning pipeline from these 2 fantastic scientists. Jamie Catalano, she actually has a paper out in bioRxiv, and hopefully she'll have a paper coming out soon. Amazing scientists, and an undergraduate who's working with me, Eve Glenn. And so what we're doing is we're looking at perforating features that we can get from the videos of these flies. Some perforating features we're really interested in are velocity, distance to wall, the magnitude of velocity. And we want to compare these behaviors between escalating, de-escalating, and stable drinking flies, and also between alcohol and natural rewards like apple cider vinegar. And then another added benefit of using this machine learning pipeline is we can also look at behaviors that are classified by combining several perforating features, including behaviors like pausing, advancing, retreating, thrashing, which from Jamie's data set from her open bar assay, she's shown that these behaviors seem to be really good markers for looking at motivational behaviors for ethanol. So, so where I'm at right now is I'm actually currently learning how to construct crosses where I can use the split Gal4 UAS system to investigate the necessity of particular subcompartments or cells in subcompartments of the mushroom body and examine what are the neurons required for the expression of escalation of drug self-administration. And so currently working on that, collecting data. It's going to be cool. And my goal after that is then to take that data, and if we find neural populations that are required, that are necessary for escalation of drug self-administration, I'm really interested in looking at what are the calcium dynamics in those compartments. What are the signaling pathways that are going on that make an alcohol escalating fly different from an alcohol de-escalating fly. And I'm really excited about this because I think we can really get at, because of the well-characterized nervous system of Drosophila, we can really get at some of these fundamental questions of what causes and what are the underlying mechanisms for an animal to self-administer more and more drugs. So that is it. My Twitter handle is DentateGyro1, and I just want to put a plug in for, I am an executive board member of Muse Mentorship, where we are trying to provide mentorship and resources for all underrepresented individuals. So hit us up if you're interested, and thank you for this time.
Awesome. Great talk. Thank you, John. If anyone has any questions, you can put them in the chat. Or unmute yourself if you'd like. And while people type their questions, John, I have a question.
Yes.
So I love your duration of bouts, average duration of bouts in your escalators. And I'm wondering if you've looked at those across the session to see how they might change.
Yes, so I actually have that data. I haven't. So what I did do was look at escalators. And so what I did find is that, and it's tough because I have only, like 22 flies, but I do notice that some flies have kind of a stable bout duration, where some flies will kind of have a consistent bout duration across the session. And then I have some that they just binge a lot in the very beginning, and then they will still go back and self-administer, but it decreases in duration. So I'm interested in this, but I think the N size is going to make it hard to, really get at what that means. But yes, I have looked at that data.
Awesome. We also have another question is, what does 50% ethanol do to general activity versus 3% or natural reward?
Right. So as far as I can tell, 50% ethanol is pharmacologically active. So if flies are exposed to 50% ethanol for sufficient amount of time, they'll exhibit kind of a hyperactivity. Some flies display thrashing and generalized, locomotion deficits. 3% ethanol does not seem to get them drunk or intoxicated. So we don't see that behavior. And that's also true for apple cider vinegar as well. We don't see that same kind of, it's hard for me to coin it. I don't want to say like aversion related, but it's almost feeling the pharmacological effects of alcohol. We don't see those behaviors for apple cider vinegar or 3% ethanol.
Excellent. Well, if there are any other questions, maybe John, just check the chat, or you can contact him via his Twitter handle. And our last speaker of the day, I'm excited to introduce. So, so I should also say that I will do my very best to pronounce this. And if I mispronounce your name or the pill you're providing, please spend your first minute correcting me. So I'm going to introduce Heloy Zhang, who's a graduate student in Gang's lab in Jinan University in China. And he'll be speaking about some really interesting differential effects of Yuizhu on Krebs signaling and neural activity across different strands of mice. So my apologies if I mispronounced that. Now you have the floor to correct me.
Thank you, Christine. Hello, everyone. It's my pleasure to meet you. My name is Hello Zhang. I'm come from Jinan University, Guangzhou, China. Today, my study is about the immediate and persistent antidepressant-like activity of Yuezhi pill in BOB-C but not B6 mouse strain, an electrophysiological and related Krebs signaling study. We all know that depression and its pharmacology treatment are associated with multiple factors, including genetics. Increasing number of studies found that ketamine produced rapid and long-lasting antidepressant-like effect both in patients and rodents, which depend on upregulating BDNF expression in brain regions. Based on this, can traditional Chinese medicine produce the rapid antidepressant effect? We found that traditional Chinese medicine formulated by Dr. Zhu Danxi 800 years ago consists of 5 herbs, which was usually used to treat the psychiatric disorders, including depression. Previous study found that yuezhi can produce the rapid antidepressant effect in mouse, which was also depend on upregulating BDNF expression in hippocampus. Furthermore, in clinical study, we also found that yuezhi can produce the rapid and long-lasting antidepressant effect in patients. We also revealed that between 2 outbreak strain mouse, including Kunming or ICR, was similar immediate antidepressant-like activities of a single dose of yuezhi or ketamine, but the persistent activity was different between strains. Strains depend PK crab signaling regulates long-lasting antidepressant response of yuezhi. Here we assessed the difference in antidepressant-like effect of yuezhi in 2 common inbred strains, including BC or B6, and investigated the underlying PK crab signaling and related electrophysiological basis. Based on that, we found that a single dose of yuezhi produced the immediate and persistent antidepressant-like effect only in BC strain mice, but not in B6J mice, both at 60 minutes and 24 hours after a single dose of administration. Moreover, we tested the expression of BDNF in hippocampus. We also found that BDNF protein expression rapidly increased in hippocampus of BC mice at 30 minutes and 24 hours after a single dose of yuezhi without alteration in B6J mice. Furthermore, we also tested the upregulation and downstream protein of BDNF. We found that PK crab signaling that regulates BDNF and synaptic protein PSD95 displayed the similar strain-dependent expression pattern, both at 30 minutes and 24 hours after a single dose of yuezhi. We all know long-term potentiation is associated with synaptic plasticity. Based on it, we measured the long-term potentiation in hippocampus after a single dose of yuezhi in both strains. We found that LTP was enhanced only in BC mice or not in B6 mice, both at 30 minutes or 24 hours after a single dose of yuezhi. Finally, mice treated with H89 are PK antagonists before or after yuezhi treatment, and the effect of H89 was also tested on ketamine. We found that the immediate and persistent antidepressant of yuezhi was blunted regardless of the time of H89 treatment. In contrast, pre-treatment of H89 only blunted the persistent but not immediate antidepressant of ketamine. Summary: the present study suggests that the strain-dependent, immediate, and persistent antidepressant-like effect of yuezhi involved in activating PK crab signaling and subsequently LTP. Initial activation of PK crab signaling regulates maintenance of ketamine's antidepressant activity. Thank you for my teacher, Professor Gangchen. Thank you for my teammates. Thank you for IBANGS to give me the opportunity. Thank you for everyone.
That was fantastic. And thank you also for joining us in the middle of the night. So see if anyone has any questions. You can put them in the chat or unmute yourself.
I have one question. Have you had, so there are some interesting substrain differences. BALB-CJ is what you studied, I assume, although the particular vendor may be important. But there's also BALB-C-Bailey-J, and I believe that Cameron and others have studied that. If it turned out there was a difference in the response to you, however you say it, to this natural substance, it'd be quite possible to map the causal gene.
We previous study, we found that the difference between outbreak strain mouse and on the treatment of a single dose of yueju, which depend on PK crab signaling. So we can focus on the increased strange. We just choose BC mice and B6J mice, and to investigate the underlying mechanism, and found that PK crab signaling was involved in the immediate.
That's quite my question. My question is really just it's a question plus an opportunity for you, just to look at substrains of BALB-C, because if you get differences like you have between BALB-CJ and B6, it would be amenable for genetic dissection.
I can't investigate the genetic dissection, and I will to investigate it in next study. Thank you.
Any other questions?